Hello, my name is Monica, and today I'm going to tell about the myosin protein. The main aim of this project is to express my vector by using the myosin protein, and I have taken my protein as myosin, gene as my gene as a NYH7 protein, and I have chosen a vector for mammalians. So here I have chosen PCMV tag 2B as my vector, and the main functions of myosin is that myosins are actin-based motor molecules with ATPase activity essential for muscle contraction, and these myosins are the proteins best known for their roles in muscle contraction, and they are wide in range for their motility process in eukaryotes. And in this next slide, we can see the main goal of this project. It is the insertion of this MYH7 gene into HEK293 cells of the plasmid PCMV tag2B vector using CMV promoter, and this MYH7 is a major gene that encodes the beta myosin heavy chains, beta MHC subunit of the cardiac myosin. And in the next slide, we can see the strategy. So here I took the gene sequence, and by using the restriction enzymes of the PCMV tag2, we got two primers, the forward primer and the reverse primers for the gene. And here we use PCR to amplify the gene of interest, and by using it, specific primers were designed which allowed the amplification. So, first I took a PCR tube with a gene sample followed by PCR amplification, and then we have to take 4 PCR tubes with tag DNA polymerase, then buffer, and with MgCl2 and water, and then we have to amplify it. After amplification, we have to analyze the PCR product on 1.1 agarose gel and visualize the bands using a DNA strain, which produces nearly 4,300 base pairs of this plasmid DNA. And then we have to do a quick gel extraction from this agarose gel, and we have to ligate the isolated restricted products using this plasmid DNA. And then we have to do the mammalian transformation. And here we are taking the HEK293 cells in the E. coli, and we have to store it in the eyes. Then we have to grow this E. coli in an exponential phase. Then we have to centrifugate the E. coli to get the bacterial cell pellet. And then we have to add the ligated plasmid to the vector, which contains the MYH7, and we have to mix it, and then we have to incubate it for 2 to 3 minutes again, and you have to give a heat shock to the cell vector, make sure at 42 degrees centigrade, and it should be followed by an immediate ice cooling. And then we are going to add the medium. I have taken Delbecq modified Eagle medium to this cell E. coli strain, and then we have to mix it, and then we have to take 50 microliters of this medium, and we have to spread it on the agarose gel, and you have to incubate it at 37 degrees centigrade for overnight or for 24 to 48 hours. In the next day, you have to pick a few colonies from the strain, like you will grow the colonies, right? So when the colonies are grown, you have to pick a few colonies, and then we have to isolate the plasmid DNA using GeneJet plasmid. So what we are doing here is a quality control validation. So we are doing it from the grown back mammalian culture, and after like isolating the plasmid DNA, we have to purify this plasmid DNA using the same thermogene jet spin column, and then you have to digest it with CMV promoter using the arcut smart buffer, and you have to spread it on the 1.1% agarose chill, and see you can identify the base pairs of the plasmid, like it would be nearly 4,324 base pairs, and here you can see the Sanger sequence for the same vector. And in the final step, we are going to determine the cells of the um, vector, and you have to determine, we can determine it by the neomycin required for the HEK293 cell selection, and it is done by lipofiction for using like HEK293 cells, and by using neomycin selection, cells were able to effectively internalize the shuttle vector, and you can see the HEK293 cells would produce the GFP protein, which would emit green fluorescence when it is exposed to the appropriate excitation light. And in the end, uh, I can say that this myosin protein affects the hypertrophic cardiomyopathy and left ventricular non-compaction and the restrictive cardiomyopathy. It leads to these diseases. So that's it. So here I have shown my expression vector, how I transformed it to my expression. And yes, that's it. Here is the end protein I got after the mutation. Yeah, thank you.